This recording is going to be an introduction to acid-base balance and also address chemical buffer systems involved in the regulation of pH. A separate recording will cover the respiratory compensation as well as the renal compensation for acid-base balance, and then a final recording will cover specific acid-base disorders. So we'll start with the importance of regulating your body pH. Our body pH is regulated very tightly to keep it in a range between 7.35 and 7.45. What would happen is if your pH gets too high or too low, it will affect a number of different body systems because it can affect the charges on say proteins, amino acids, which can affect the function of proteins, and so a lot of body systems will not work properly if you start getting outside of that range. Now what I want to bring your attention to though is we do have this pH between 7.35 and 7.45. They'll say like the average is you'll see a lot of times pH 7.4. Now the differences that you see is this here, the 7.35 is what we find in venous blood. And that has to do with the transport of carbon dioxide. Here is what we would see in arterial blood. Now what you'll notice is, if you remember any biology, is that you learn that pH of 7 is neutral. Anything above 7 is basic, or they refer the term as alkaline. Anything below 7 is acidic. The thing is, when we use the terms acidosis or alkalosis in talking about individuals with acid-base disorders, is we're indicating if the pH, say, is above 7.45, that person is said to have being alkalotic. If the person's pH is less than 7.35, they're considered acidotic. And you notice they're not happy because things aren't going to work very well. So we try to keep that pH in this very narrow range. And I'm specifically referring to the pH of your extracellular fluid. Now we always are dealing with, most of the time, dealing with producing a lot of acids in our body. That's typically what we have to deal with the most, but there are instances that we're going to have to deal with bases. But I just want to bring your attention to all the different acids that we do produce in the body and differentiate between fixed acids. These are going to be fixed acids. They don't readily leave solution, and so they'll have to be dealt with by removal at the level of the kidneys. And then we have over here, we're going to have an example of a volatile acid, which can leave solution readily. And the reason why the one that I'm going to show you is a volatile acid, because it involves carbon dioxide. From aerobic respiration of glucose, we do produce carbon dioxide. And I'm going to write that here. But the acid, you're like, there's no hydrogen ions here. Well, carbon dioxide can combine with water to form carbonic acid. So carbonic acid can dissociate into hydrogen ions and bicarb. And actually, I'm going to write this equation. You're going to see this equation many a time. Carbon dioxide can combine with water to form carbonic acid, which then can readily dissociate into hydrogen ions and bicarb, and vice versa. It can go in either of the directions. So this one's considered a volatile acid. The other ones are considered fixed acids. Well, examples. Anaerobic respiration of glucose, you can produce lactic acid. Oxidation of sulfur-containing amino acids, like something like cysteine, you can form sulfuric acid. I'm going to jump to the end, is hydrolysis of nucleic acids or metabolism of nucleic acids and phosphate-containing proteins. You get phosphoric acid. And the last one, if you have incomplete oxidation of fatty acids, you can produce substances that are referred to as ketone bodies. And I'm just going to write that for now. You're going to see the name of one of them shortly. Oops, sorry, not ketone acids. They're called ketone bodies. Ketone bodies are weak acids, and they include something called acetoacetic acid and beta-hydroxybutyric acid. All of these are acids that we're going to have to deal with. If you accumulate too many of these acids, your pH will drop and you won't be happy. So we'll have to deal with them. Well, one of the things I do want to show you, which was kind of briefly kind of insinuated, is there's this relation between carbon dioxide levels and your pH. And because of that reaction, if I go back here, this reaction that you saw here is the more carbon dioxide that you have, you'll actually, this equation goes in this direction, you'll have a lot more hydrogen ions. And so the higher the carbon dioxide levels, the higher the hydrogen ions, the lower the pH. Because pH is equal to the negative log of the concentration of hydrogen ions. So the more hydrogen ions you have, the lower the pH. And then if CO2 levels fall, you're going to have less hydrogen ions and your pH will be greater. So there's this inverse relationship between carbon dioxide and plasma pH. The higher the CO2 levels, the lower the pH. And that will become important. You're going to see this equation about the relationship between carbon dioxide and hydrogen ions over and over and over again. Now, a kind of quick thing about acids and bases, because they're going to play an important role in understanding how buffers work, is an acid is a substance that can donate a hydrogen ion or release a hydrogen ion. Some people, I'm old school, I always remember acids are proton donors. They're willing to give up a hydrogen ion because this actually is a proton. And bases are substances that can accept this proton. So they're proton acceptors. And we can also distinguish between acids or bases as being strong or weak and how readily they dissociate. So here, hydrochloric acid is a strong acid. If you put that in water, it will completely dissociate into hydrogen ions and chloride. And this is going to cause that pH to drop quite a bit. Okay? A weaker acid won't. Okay? And I'll show you a picture and I'll come back to that base. So here's a strong we put a strong acid in solution. It's completely dissociated. Okay? Completely dissociated. Here's showing you a weak acid. It's not complete. I've given up a proton here, but a lot of them are still bound. So weak acids don't readily dissociate as much. So they're not going to change the pH quite as much. And using a weak acid and what I call the conjugate base, I'm going to show it to you in a second, is that's going to be good in creating of buffers. So just kind of keep that in mind. So I'm just going to go back to this picture where you see here's bicarbonate, which is a base. It's willing to take up a hydrogen ion. And so what you see here is this combined with the hydrogen ion and you formed carbonic acid. Carbonic acid is what it's going to be considered the acid, and that actually is not a strong acid. It's actually a relatively weak acid. So it won't change the pH quite as dramatically. So what I wanted to mention is there are 3 different mechanisms that we have in place to regulate hydrogen ion concentration, to control pH levels. The one that works the quickest are chemical buffers, and that's what we're going to address most at the remaining of this recording. This acts very quickly. These buffer systems prevent drastic changes in pH to kind of prevent any damage to tissues and cells, and it will work until we can get rid of them using either of the other two systems that we have, which are physiologic buffer systems. We have the second to act is respiratory. That works relatively quickly, but not as quickly as the chemical buffer systems. And then the last takes a little bit longer to act and to get fully ramped up and working is the renal buffer systems. But this is extremely important, is the renal buffer systems. And, or sorry, the renal, should say compensatory system. So the chemical buffers are what I want to talk about. So we have these buffer systems that are in place to prevent drastic changes in pH of either your intercellular fluid or your extracellular fluid. And so you see some examples written for you here. So let's talk, actually here, let's talk about, before I do that, is when you have a buffer system, a chemical buffer system, it includes I'll say a weak acid and its conjugate base. So conjugare is a Latin word that means to join together, because that base will conjugate with a hydrogen ion to form the weak acid. Or you have a weak base and its conjugate acid. And so we have a mixture of an acid and base that are components of our buffer system. So let's figure out how it works. So the first system we have is the carbonic acid bicarbonate buffer system. Hands down the most important one in regulating pH in the extracellular fluid, but it does have some limitations. So we'll mention those limitations in a moment. So with this buffer system, we have our acid, which is carbonic acid, which actually is directly related to CO2 levels. And we have the base is right here. So what happens, for example, you have added fixed acid into solution. I want to tie up those hydrogen ions, otherwise the pH is going to start to drop drastically and affect how things function. So what do I have is I've got the bicarbonate. So the bicarbonate will join up with this hydrogen ion. What's going to happen is you create carbonic acid, and then that dissociates into carbon dioxide and water fairly readily. One of the things, though, we have to worry about is I have to make sure I have enough bicarbonate. I have to have a bicarbonate reserve. So it will be limited if all the bicarbonate is tied up. So something that we always have to worry about. Another thing we have to worry about is you have to have proper functioning of your respiratory system and the respiratory centers, because I'm going to have to get rid of this carbon dioxide. And so I'm going to need to get rid of that. Now, another thing I have to worry about, which limits this buffer system, is this buffer system will not be affected or effective in regulating the pH in your extracellular fluid if the pH change was due to elevated or depressed CO2 levels, because you can't use your own acid as a buffer, or sorry, let's reword what I just said. A buffer system can't protect against the changes in the concentration of its own weak acid, and technically CO2 is the weak acid because it'll combine with water and form the carbonic acid. So this buffer system won't be is important in certain acid-base disorders that we talk about later, but it's going to be very important in others. Okay, so we'll just leave it at that for now. So that gives you one example of how this buffer system can work. Other buffer systems that we have is we have a phosphate buffer system, which works very well inside cells because you have a lot more phosphate. Now in this buffer system, right here, this is one component. Right here is another component. This is monohydrogen phosphate. This is dihydrogen phosphate. The dihydrogen phosphate is the acid. The monohydrogen phosphate is the base. And what you're seeing on the picture is you see a hydrogen ion that was created in the cell. The monohydrogen phosphate combined with that hydrogen ion and formed dihydrogen phosphate. Dihydrogen phosphate is a weaker acid than straight off hydrogen ions. So what we do with a buffer system is you're going to convert a strong acid into a weaker acid, or a stronger base into a weaker base. So in this case, a strong acid into a weaker acid. And what you'll notice too is, where did that hydrogen ion come from? Well, if you look carefully, is you'll notice over here, you see fatty acids, you see this hydrogen ion, and you see acetoacetate. Well, I had mentioned previously the incomplete oxidation of fatty acids can generate acetoacetic acid, which is a ketone body. Acetoacetic acid dissociates into acetoacetate and a hydrogen ion. So that's where I'm going to have to deal with. I'm going to need some sort of buffer to kind of protect that change in pH that can result from the acetoacetic acid dissociate. Now that can dissociate inside here, the liver cell, but it also can happen out here in the extracellular fluid. And you'll notice out here, this bicarb is real important in buffering the extracellular fluid. So you see, if that acetoacetic acid was in the blood and it dissociates into the hydrogen ion and acetoacetate, that bicarbonate will buffer it. Okay, so you can see that there. A use of the the phosphate buffer system is also, we'll see it's important as a urinary buffer. So in this scenario, a hydrogen ion is being secreted into the tubular lumen, and it's going to combine with that monohydrogen phosphate to form dihydrogen phosphate. That will keep the pH of the urine from dropping too drastically, which can be very uncomfortable, and also allows us to be able to secrete a lot more hydrogen ions when we do that. Now we also have, I go back to this picture where you see that PR is, we have, the PR stands for protein, is we do have protein buffer systems. But a example of which actually even more protein buffer system that's even more important is in red blood cells, we have hemoglobin. So we'll come back to that in a second. So let's just going to talk about the protein buffers. So proteins are made up of a number of different amino acids, and at one end of the protein we have a carboxyl group, and in the other end we have an amino group. Well, what you see here is what we tend to see at physiologic pH, is the carboxyl group has dissociated and the hydrogen ion has been released, and you see the charge like this. The amino group is actually picked up, here's an amino group, has picked up a proton, and so it's positively charged. But what can happen is that the pH in your body starts to change, is this carboxyl group now can combine with a hydrogen ion and form this, and this end can release the hydrogen ion and form that. So the proteins can act as buffers because they have these groups that can act as acids or bases. And the term that they use for the fact that proteins, or I should say even amino acids, can act as chemical buffers or acids and bases, is this term amphoteric. They can act as acids or bases. Now the protein buffers won't, the normal protein buffers kind of are slow in transporting hydrogen ions across the cell membrane. So they can't really protect against very large changes in pH because these proteins, most of these proteins are inside cells. The hemoglobin buffer system is one though that can buffer very immediately, very quickly. And the hemoglobin buffer system is found in our red blood cells. And you can see the little bit of picture here is hemoglobin. And the globin change in hemoglobin is they have histidine residues. And the histidines can act combine with hydrogen ions and help prevent drastic changes in pH. So hemoglobin is an example of a protein buffer system, but it's specific for red blood cells. So I'm going to stop here with the buffer systems. Next recording, we're going to look at how the changes in ventilation can regulate pH, as well as how the kidneys can work.